Hey guys, this week we'll be talking about cellular respiration, and we will be going over, or we will be using the concepts of chemistry and thermodynamics that we covered the last 2 weeks to help us better understand how cellular respiration works. Before we start the lecture though, I just want to remind you guys of some of the different chemistry concepts. So accepting electrons creates a negative charge. Losing an electron creates a positive charge. Chemical bonds contain a lot of energy, and when those chemical bonds are broken, a lot of energy is released. Some of that energy, though, will be lost as heat during chemical reactions. And the buildup of molecules creates a concentration gradient and creates a lot of energy. And that energy will be harvested as ATP so that our cells can use that ATP for its chemical processes. So we all know that everyone breathes in oxygen and breathes out carbon dioxide. But why is that exactly? Well, the reason, obviously enough, is cellular respiration. We breathe in oxygen to use for cellular respiration, which is one of our reactants, and then we breathe out a product of that reaction, which is carbon dioxide. We excrete the water that is produced from from cellular respiration through our urine, and then of course we get our other reactant, glucose, from the food that we eat. Cellular respiration happens in a little organelle called the mitochondria. Mitochondria can be found in both plant and animal cells, so therefore both of these type of organisms can undergo cellular respiration. Mitochondria is a double membrane bound organelle. The outer membrane covers the entire mitochondria, while the inner membrane folds in on itself to create something called cristae. The inside of the inner membrane is going to be called, the inside of the mitochondria is going to be called the matrix, and then in between the two membranes is called the intermembrane space. The first step of cellular reaction will happen on the outside of the mitochondria, while the last three steps of cellular respiration will happen on the inside of the mitochondria. So let's just go over the main characters of cellular respiration so that you guys can better understand cellular respiration. So we're going to be looking at glucose, which is a 6-carbon molecule. We're going to be looking at hydrogen, which is, since there's a positive charge, it means that it has no electrons and is just a proton. There's going to be electrons, which of course has a negative charge. And then there are these coenzymes, NAD+ and FAD+, that are going to be like carriers for the hydrogen ions from the first and third step of cellular respiration, and is going to be very important in the last step of cellular respiration, where most of our ATP is going to be generated. So cellular respiration is called a reduction-oxidation reaction, or for short, a redox reaction. During redox reactions, hydrogen ions are taken from one molecule and given to another. If hydrogen is removed from a molecule, we say that the molecule is oxidized. And then if the hydrogen is given to a molecule, we say that that molecule was reduced. So glucose is oxidized, and then the hydrogen ions are given to the oxygen. This turns the glucose into carbon dioxide, and then the oxygen into water. If the oxygen, so the reason why it is called a redox reaction is that if the oxygen state of an element increases, we say that it was oxidized, and then if the oxygenation state was decreased, we say that it was reduced. One easy way to remember this is that it is a reduction of charity if you receive something. So like I was saying earlier, there are 4 steps for cellular respiration. Our first step is glycolysis, and it happens on the outside of the mitochondria. 2 ATP will be generated in this step. If there is oxygen present, then our pyruvate molecules will move into the mitochondria, where it will undergo the preparatory reactions, where it will prepare the pyruvate to enter into the citric acid cycle. And then the biggest step in cellular respiration, though, is the electron transport chain. Because all of the hydrogen ions that were taken from glycolysis, the prep cycle, and the citric acid cycle will be taken to the electron transport chain, where most of our ATP will be generated. Notice here how we have some carbon dioxides being released during cellular respiration. All of our 6 carbons that we started off with in glucose will be released as carbon dioxide throughout some point of the entire process. So let's go over glycolysis first. So glycolysis is when glucose is broken down into 2 pyruvate molecules. It has 2 steps and involves 10 enzymes. But don't worry, I'm not going to make you guys memorize all those enzymes because it would just be insane. But we can still break it down into 2 steps to help make it a little bit more understandable. The first step is called the energy investment step because it requires energy in the form of ATP to start. The second step is called the energy harvesting step, where ATP is going to be produced. So during the energy investment step, our glucose molecule, which again contains 6 carbons, is going to receive 2 phosphate groups from our 2 ATP. The energy that is given to the glucose molecule by the addition of these 2 phosphates is going to break that glucose molecule in half to generate 2 G3P molecules. We call this the energy investment step since we have to use some ATP to start off our reaction. Our second reaction, though, produces ATP, and it produces enough ATP where we actually have a net gain of ATP, so that glycolysis itself doesn't feel like it's a worthless step. So during our second step, our 2 G3P molecules goes through a series of reactions where it will eventually become 2 pyruvate molecules. So we start off with 1 glucose molecule, and then we'll end up with 2 pyruvate molecules. Those pyruvate molecules will go through a series of reactions where 2 hydrogen ions will be taken from the 2 G3P, which will then be picked up by the coenzyme NAD, which then becomes NADH. So at the very end of glycolysis, we'll have 2 NADHs and 4 ATP that would have been developed by substrate level ATP synthesis. And what this means is that enzymes were able to use the G3P at some point to produce 4 ATP molecules. And because we used 2 ATP to start off glycolysis and we produced 4, we now have a net gain of 2 ATP through the entire process. At the end of glycolysis, we end up with 2 NADH, 2 pyruvate molecules, and a net gain of 2 ATP. Glycolysis is the only step of cellular respiration that does not need oxygen, which is really important. The other 3 steps of cellular respiration does require oxygen. If there is oxygen present, then the pyruvate molecules will enter into the mitochondria to undergo those other 3 steps of cellular respiration. If there is no oxygen, then those pyruvate molecules will stay within the cytoplasm of the cell and undergo fermentation. So fermentation is when the pyruvate molecules from a glycolysis receives hydrogen ions from NADH. This then turns pyruvate into lactate, which within the watery matrix of the cytoplasm turns into lactic acid. This is really important because it frees up those NADHs to go and be used again within glycolysis, so that ATP can continue to be produced, even if it is just a little. So without fermentation or oxygen, glycolysis wouldn't be able to occur, and no ATP would be produced within our cells. So if you were doing a really, really heavy workout and you were really out of breath, your muscles would just stop working if it wasn't for fermentation. When you exercise for long periods of time and you run out of breath, your muscle cells can no longer perform cellular respiration and instead performs fermentation. This gives you just a little burst of energy as you work out, but it is not obviously sustainable. So when you are done exercising and out of breath, your body causes you to breathe really fast so that you regain the needed amount of oxygen in your blood, so your cells can perform cellular respiration again. The lactic acid builds up in your muscles as you exercise, which is that burning sensation that you feel during your workout. The soreness you feel in your muscles the next day is the anti-inflammatory agents of your immune system working on those damaged cells to heal them. When your blood oxygen levels return to normal, the blood carries away the lactate to the liver, where it can be converted back into pyruvate and then enter into the rest of the cellular respiration process. Or if your body doesn't need ATP at that time, it can be converted into glucose and stored as glycogen in the liver. Fermentation that happens within microorganisms like yeast and bacteria can be used to make really great products like bread, wine, and beer. This process of fermentation is going to be a little bit different though. Instead of lactate, ethyl alcohol and carbon dioxide are produced. In bread, the carbon dioxide is what causes the bread to rise, and then the ethyl alcohol is what turns grapes into wine and wort into beer. So when used by animal cells, fermentation is going to cause two lactate molecules, and then we're also going to get two NAD pluses that can then be used back in glycolysis. But when used within bacteria and fungi, we're instead going to get two ethyl alcohol molecules, two carbon dioxide molecules, and we are still going to get those two NAD pluses. Glycolysis and fermentation are the energy pathways that do not require oxygen. We call this anaerobic respiration. With glycolysis, you will produce a net gain of 2 ATP. Fermentation doesn't produce ATP, but it's still really important since it frees up those coenzyme NAD pluses to go be used in glycolysis again so that our bodies can keep regenerating that ATP. So we call fermentation and glycolysis anaerobic respiration. If there is oxygen present though, that pyruvate molecule will move into the matrix where it will undergo aerobic respiration. Air sounds like oxygen, so that should help you to remember aerobic respiration. The second and the third step of cellular respiration will happen within the matrix of the mitochondria, and then that fourth step of cellular respiration will happen along that inner membrane. Some of the parts of the step will happen on the inside of the inner membrane, along the inside of the inner membrane, and then in the intermembrane space here. So when oxygen is present, the pyruvate molecule will enter into the mitochondria and enter the second step, which is called the preparatory cycle. In this reaction, hydrogen is taken from the pyruvate molecules and given to NAD+, making NADH. This transfer produces one carbon dioxide molecule. The result is a 2 carbon dioxide molecule called acetyl that is attached to an enzyme called coenzyme A. So together, it's going to form a molecule called acetyl-CoA. Because 2 pyruvate molecules entered into the preparatory cycle, The preparatory cycle will happen twice per glucose molecule. At the end of the preparatory cycle, there will be 2 acetyl-CoAs and 2 NADHs. So we started off with a 6-carbon molecule, and now we are down to only 4 carbons. 2 of the carbons were released as carbon dioxide during the preparatory reactions. Our third step is called the citric acid cycle. Those 2 acetyl-CoAs enter into this third step, which also occurs within the matrix of the mitochondria. The first thing that happens in this cycle is that the acetyl is transferred to another molecule, producing citric acid, which is a 6-carbon molecule. This then frees up that enzyme CoA to go back to the preparatory cycle to collect more acetyl molecules. The citric acid is then oxidized a few times, which produces NADH, FADH2, and carbon dioxide. An ATP is produced by substrate level ATP synthesis, as it was in glycolysis. So per glucose molecule, at the end of the citric acid cycle, we will have 4 carbon dioxides, 6 NADHs, 2 FADH2, and then 2 ATP. So here we have the entire process of the citric acid cycle. So we have our acetyl-CoA right here, which will attach to a 4-carbon molecule, making a 6-carbon molecule. It will be oxidized twice, which releases 2 NADHs here, and then 2 carbon dioxide molecules. So we end up with a 4-carbon molecule again. And ATP will then be produced through ATP synthesis, or substrate level ATP synthesis. Our 4-carbon molecule will undergo a redox reaction 2 more times, producing one more FADH2 and then 1 more NADH. We are then left with our original 4-carbon molecule, so that the entire process can happen again. So per glucose molecule, at the end of the citric acid cycle, we have produced 4 carbon dioxides, 6 NADHs, 2 FADH2, and then 2 ATP. So we no longer have any carbons from our original glucose molecule. So then all of the coenzymes that we have that have been produced throughout those first 3 steps will then go to the 4th step, which is called the electron transport chain. These coenzymes will drop off those hydrogen ions and the electrons and are then going to be released to go pick up more hydrogen ions and electrons from our first 3 steps. The electron transport chain consists primarily of the electrons passing through a bunch of carrier proteins that are located within that inner membrane. As electrons move through the carriers, the electron's energy is formed into ATP. So we have 2 NADH from glycolysis, 2 NADH from the prep cycle, and then 6 NADH and 2 FADH2 from the citric acid cycle. Roughly about 3 ATP can be made from each NADH, and 2 ATP can be made per FADH2 coenzyme. This amounts to a total of 36 to 38 ATP from 1 glucose molecule. So this is going to be the primary step in cellular respiration, where we're going to get the majority of our ATP for our processes in the cell. So here we have our enzymes located within that inner membrane of our mitochondria. The NADH and FADH2 will drop off its electrons and hydrogen ions here, so that they're now freed up to go pick up more hydrogen ions from our previous steps in cellular respiration. As the electrons flow through these enzymes, it will cause the hydrogen ions to flow into that inner membrane space. This creates a concentration gradient of hydrogen ions, which naturally causes the hydrogen ions to want to flow back into the matrix to establish equilibrium. As it relates to thermodynamics, the energy created by the movement of the electrons decreases the entropy of the cell, since all the hydrogen ions are within that inner membrane space. This decrease in entropy, though, takes a lot of energy. Another way of saying it is that the buildup of hydrogen ion creates a lot of energy. Notice that the NADH drops off the electrons and hydrogen ions at that first carrier, while the FADH2 drops it off just right before the second enzyme. That's why NADH is able to produce one more ATP than the FADH2, since its electrons will flow through more enzymes than the FADH2. Since it is the natural tendency of the cell to increase its entropy, those hydrogen ions will flow back into the matrix of the mitochondria to reach equilibrium. They flow into the matrix through an enzyme called ATP synthase, which is also located within that inner membrane. The energy caused from the hydrogen gradient transfers that energy to the ATP synthase, which then allows it to create a lot of ATP. So here you guys can see the entire process. We have our electrons flowing through our enzymes, which that energy is what allows the hydrogen ions to flow through that into the inner membrane space, creating our concentration gradient. Since there are more hydrogen ions located on the inner membrane space, its natural tendency is going to flow back into the matrix until there is an even amount of hydrogen ions on either side of the membrane. As the hydrogen ions flow through the ATP synthase, the ATP synthase will then generate a lot of ATP. So from one glucose molecule, we have generated a maximum of 38 ATP. 2 from glycolysis, 2 from the citric acid cycle, and 34 from the electron transport chain. Although this is a lot of ATP, this only represents about 40% of the available energy from our glucose molecule. The rest is lost as heat, as stated from that second law of thermodynamics. There are some cells, though, that do not even generate this much ATP. These produce only 36 or less ATP per molecule. So NADH can produce 3 ATP. Since there are 10 NADH, they will produce a total of 30 ATP. FADH2 produces 2 ATP, and since there are 2 FADH2s, we can produce 4 ATP from that. So this will give us a minimum of 34 ATP that we can produce from cellular respiration. So as we talked about a couple of weeks ago, there are several biological molecules in the food that we consume, those being proteins, carbohydrates, nucleic acids, and lipids. Besides carbohydrates, proteins and the fats that we consume are able to be broken down into its smaller molecules and enter cellular respiration at different points. One of the lipids that we talked about was triglycerides, which are made up of 1 glycerol molecule and 3 fatty acid tails. Fatty acids can enter into the cellular respiration during the citric acid cycle, and the glycerol group can enter into the very first step of cellular respiration since it is a carbohydrate. The fatty acid has 18 carbons within its chain, so it can generate 3 times the amount of acetyl groups as glucose can. That is why fat has so much more calories or energy than sugar does. When our liver is short of glycogen, our body is still in need, and our body is still in need of ATP. Our liver can break down proteins to help supply this energy. The only part of the protein that can enter into cellular respiration, though, is the hydrocarbon backbone. The amino groups will be first removed and then turned into urea, and then this frees up that hydrocarbon chain to enter into cellular respiration. The amount of energy that that particular protein can produce will depend on the amount of carbons found within that hydrocarbon chain. So just to recap, we have 4 different steps in cellular respiration, that being glycolysis, which produces pyruvate, the Krebs cycle, which produces acetyl-CoA, the citric acid cycle, which produces a lot of NADH and FADH2, and then the electron transport chain, which produces most of our ATP. The main source of energy that our bodies will use for ATP is carbohydrates, but the other biological molecules that we consume, primarily proteins and fatty acids, can also be used as an energy source if there is no glucose available. Proteins can enter into any step of the cellular respiration. Carbohydrates can go through the entire cycle. The glycerol molecule from our fatty acids can enter into glycolysis, and then our fatty acid tails are able to enter into the citric acid cycle. Any of those intermediary molecules, like pyruvate and acetyl-CoA, are able to be made into proteins, carbohydrates, and fatty acids if there are too many of them. As a result, we are able to store any of those extra molecules in case we need that energy for later. This is why we will gain a lot of weight if we eat too much food, especially during the holidays. So thank you for joining me, and as always, let me know if you have any questions.